great pleasure to welcome today's seminar speaker, Dr. Regina Baucom. Hope you say your name right.
That's
correctly
correct,
again. Professor in the Department of Ecology and Evolutionary Biology at the University of Michigan. Dr. Baucom is an internationally recognized leader in the evolutionary ecology and genetics, particularly in plants population facing intense human-mediated selective pressures. Her research beautifully integrates fieldwork, genomic, quantitative genetics, and ecology and evolutionary theories that I think we're going to hear a little bit about today. Much of her work centers on the common morning glory, a powerful model for understanding herbicide resistance. Dr. Baucom also has pioneered research in the ecology and evolutionary consequences of emerging herbicide technologies. In short, her work shows how contemporary evolution is happening all around us, often driven intentionally or not by human hands. Dr. Baucom's scientific impact is matched by her deep commitment to mentorship, training, and community building. She has advised dozens of graduates and undergrad students, led NSF and USDA-funded projects, served as associate chair for graduate students at Michigan, and contributed nationally through editorial leadership at the service in the Society of the Study of Evolution and the American Genetic Association. She's also a visible champion of diversity, equity, and inclusion, co-creating the Diversify EEB resource. If you don't know what it is, you should look on about it, especially if you are a woman in science. Her work has appeared in leading journals, including Evolution, New Phytologist, Molecular Ecology, and Evolution Letters, society journals, as per her philosophy, sparkling broad public interest throughout coverage in major science outlets. So please join me in welcome and give a welcome to Dr. Regina Baucom, whose seminar today will explore from pollination to overcompensation and how sublethal herbicide drifts alter plant traits, fitness, and ecological interactions.
Wow. Thank you, Fabienne. That was really nice. Can everyone hear me? Okay. Awesome. I can put on my teaching voice as well, right? So I kind of have a small voice, but I can project it when I need to. So I want to send a shout out to the Ecology Center for inviting me to speak, and for Kathy for nominating me to speak, and Lexi and Brian for taking care of my schedule. It's been really wonderful so far. I haven't been to Logan before, so it's excellent to see mountains. We don't have mountains in Michigan, right? Michigan's very flat in comparison. So it's really wonderful to see some topography and to meet some really fun people. Right, so today I'm going to talk about a couple of different projects that we're working on in the lab that focus on using herbicide drift as a selective force. The first project that I'm going to talk about is using episodes of herbicide drift to understand if drift from the herbicide dicamba can disrupt or interrupt plant pollinator interactions. And the second project is one in which we're looking at the impacts of dicamba drift, again, the same herbicide, on a plant's ability to do better than normal, and that's called overcompensation. Okay, so to start us off, we all know that plants live in a world that's dramatically altered by humans, whether we're talking about weedy plants that exist in agricultural areas that are being exposed to fertilizers, all sorts of pesticides, or plants that can exist in urban spaces, right? So you have to have a population that can survive in an area with a lot of concrete and asphalt. Also, plants are exposed to human-mediated climate change. So humans are imposing a lot of direct selection on plant populations. And for the majority of my career, I've been interested in how plants can rapidly adapt or not to different scenarios of human-mediated selection. I focused mostly on this dynamic, and so I'm showing you a farmer who's spraying their cornfield with herbicide. And so we have a corn crop that is tolerant of the herbicide Roundup, which the active ingredient in Roundup is glyphosate. So if I'll talk about them both using glyphosate or Roundup, I mean the same thing. But basically, we've got this transgenic maize crop or corn crop. They can plant the corn, it grows, and then spray an over-the-top application of the herbicide Roundup. The crop will live, but then the weeds will die back. And that's been a really helpful technology that was introduced in the late 90s and became very widespread and wide used. But we can always, nature always finds a way, and weed species have evolved resistance to Roundup. So I've been working on the common morning glory, Ibumea purpurea, for actually the majority of since I started grad school. And you can see there's a big population of the common morning glory that's infesting the side of a soy field. It's one of the top 10 worst weeds of southeastern US row crop agriculture. As part of one of the first papers to come out of my lab, a postdoc, Adam Kuster, went to populations in the southeast and the Midwest of the common morning glory, collected seeds from those populations, and we did what's called a dose response curve, where you assess how resistant a population might be. And what we found, just sort of the broad take home, is that there are two areas of what we would call hotspots of resistance to Roundup in this species, where these populations had a high survival. Okay, so we've got multiple populations that show high survival when they get exposed to a high level of Roundup, and other populations that are still highly susceptible. So there's variation across the landscape for the level of glyphosate or Roundup resistance in this species. And one of the reasons for that could possibly be because there's a fitness cost associated with glyphosate resistance. So I'm showing you on the x-axis, it's not labeled, but that's the level of resistance across populations. And as populations become more resistant or have higher survivability, you have a lower proportion of seeds that will germinate. So our fitness cost in the species is not number of seeds, which is what we will often look at, but instead it's the ability to germinate or not. Okay. In more recent work, we've wanted to address what are the genes associated or responsible for glyphosate or Roundup resistance in this species. And so we sequenced the morning glory genome, assembled it, and looked for signs of selection. And what I'm showing you here is basically a bunch of chromosomes and the results of a bunch of different types of selective screens. And the blue arrows that you see here in this middle circle just point to regions of the genome that were showing strong signs of selection. So on chromosome 10, we had a really strong signal of selection associated with Roundup resistance in this species. And within those regions that were showing signs of selection, we found genes involved in detoxification. So we think like cytochrome P450s, glycosyltransferases. So we think that the genetic mechanism of resistance in this species is due to herbicide detoxification. We're currently in the progress of trying to develop functional genomic resources for this species. We can't CRISPR it or do anything, fun like that to figure out which particular gene is responsible, but we have at least a handful of about 50 genes that we're interested in from this survey. Okay, so that's kind of a broad overview of what I've mostly been working on in the lab, focusing on one single species and its ability to resist or not Roundup. As I was doing this work, being in a crop situation or agricultural landscape, I always focused on this one single species where I would collect seeds, from multiple populations. But what I was always impressed by is that in areas beside the crops, there's always waste spaces, we call them waste spaces, where there are plants and communities of weeds that are existing. And those weeds have communities of pollinators and herbivores and other organisms coexisting along with them. So I became interested in wondering what's happening with these weedy plant communities around agriculture that are likely getting exposed to maybe not the field dose, which is like the recommended dose for killing a plant, but a lower dose, a sublethal dose of herbicide. How does that impact plant traits? How does that impact plant-insect interactions? Okay, and so one of the reasons we might be concerned about this is because let's say a farmer is putting their herbicide down. I'm showing you a little cartoon. We've got a tractor where we're spraying our herbicide. We can have a couple different ways that herbicide can accidentally hit areas that are unintended. One is from spray drift, where you're just spraying it, the wind blows, and herbicide molecules move and hit unintended areas, whether they're non-tolerant crops or just surrounding wild vegetation. We can also have this really freaky phenomenon that can happen if the temperature or humidity is a particular, very humid, high temperatures. We can have, put the herbicide down and it can revolatilize into the air column and then move and hit areas that are unintended, so non-target areas. Okay, and so the concept of drift it's always been kind of important, but it became much more important. Oh wait, I'm going to show you what drift looks like because it's kind of fun. Okay, so this is just an example of a really major drift event. We've got red dye in an herbicide. The farmer's applying their herbicide, and you can see the wind is just taking that herbicide, and it's going to blow and hit areas that they probably didn't want to be exposed to herbicide. Okay, so drift is a problem. It's always been a problem. It became an especial problem around 2017 when the EPA approved the over-the-top application of the herbicide dicamba. Oops, I didn't want to go back there. Dicamba is a synthetic auxin herbicide. It's an old herbicide. It was developed around World War II times when a lot of pesticides were developed. And it's been used for a long time. It went out of favor when Roundup Ready technology made a splash, and people appreciated Roundup because it's not toxic. It's not immediately toxic to humans, whereas herbicides like dicamba and atrazine are moderately toxic or very toxic. And so in 2017, dicamba was approved. The other problem with it, in addition to it being moderately toxic, is that it's very drifty, right? It has a high propensity to drift, and that's always been the case with this herbicide. And so what I'm showing you here is a farmer in his field. It is a non-tolerant soy crop. At about sometime in the 2000s, Monsanto developed a dicamba-tolerant soy strain because of the evolution of Roundup resistance in weeds. And farmers were just, at the end of their ropes with particular species like Amaranthus, Morning Glory, but, mostly it's Amaranthus that's been problematic. And so they developed this glyphosate-tolerant soybean crop that people were planting in 2017 and 2018 and spraying dicamba over the top. We had a ton of dicamba drift events, and this was all in the news. So you might remember seeing it in 2017, 2018, 2019. We still have drift events that happen because farmers are continuing to use dicamba-tolerant crops, and dicamba is very drifty. So this has led to a lot of lawsuits where farmers who are not using the tolerant bean would, a farmer close by would be using it, a drift event would happen, and it would decrease the neighbor farmer's yield. In addition to lawsuits, there was even like one farmer went and shot another, right? So it's really, really contentious. And it's not in the news quite as much, but there's a bunch of lawsuits back and forth about whether or not dicamba can be used, right? So whether or not it's out there for fair use. Okay, right. So dicamba, like I said, is an old herbicide. It's a synthetic auxin. And so auxin is a really important plant hormone that's involved in most every component of plant growth and differentiation, right? There's like 500 different auxin receptor genes in Arabidopsis. Like auxin's very important. When we use auxin as an herbicide, as a synthetic auxin, It kills plants by deregulating their growth, distorting cell division, and eventually leading to plant death. Okay, so here's a pattern of use of dicamba. So we can see that starts at 1990. We can see that it's been in use. It went out of use when Roundup Ready technology sort of swept. But then you can see in 2017, 2018, it's just exponentially increased. And some of that exponential increase is not just people that want to use the dicamba tolerant soy and the dicamba as a method of weed control, but it's also that farmers were using or purchasing the dicamba tolerant seed because they wanted to make sure that their crop would not, be hurt by the neighboring farm's dicamba use. Okay, so it just kind of swept and people are using it broadly. This is a close-up of what it looks like when a soy field gets drifted with dicamba if it's not tolerant to dicamba. So we can see this typical cupping shape of the leaves. They kind of look like cups, and then they look wrinkled on the inside. So members of the Fabaceae are very sensitive to dicamba, and it's easiest to see dicamba drift events on members of the bean family, but we can see it across all plants too. Okay, so this is just to give you a sense for estimated use in 2019 of dicamba, and effectively the estimated use is wherever there's agriculture, this herbicide that's drifty is going to be used, right? So just to give you a sense of the scale of the problem of, we're using a new herbicide, this novel technology that drifts a lot and could be impacting natural organisms around crops. Okay, so I became really interested in this concept and I wanted to understand. People have been working on dicamba drift for a while, but mostly in the context of crops, right? They wanted to understand how much yield is lost when a crop gets hit with a particular dose of dicamba drift. There's less work on the natural organisms that exist in agricultural spaces. So I wanted to understand, how do weed communities respond when they get hit with this dicamba drift in terms of plant traits? Are we disrupting growth? Are we disrupting size? Are we disrupting patterns of flowering? And then also I was interested in that, but also wanted to put it in the context of understanding how changes to plant traits from being exposed to the herbicide dicamba at drift levels might alter plant pollinator interactions, and wondering, is this novel use of dicamba and its exponential increase, is that going to lead to another way that our pollinators are not getting the adequate resources that they need, right? Okay, and so what I'm showing you here is effectively just a cartoon where I'm showing you flowering day, flowering number plotted by flowering day. And I've got two curves there. I've got a control weed population, when it's going to start flowering and the number of flowers that it will produce. You can see peak flowering. And then I've got pollinator phenology in this blue curve. And so, we would hope that the pollinator life history would overlap well with the normal pattern of production of flowers by weedy plants. Well, if weeds are exposed to dicamba, typically plants that get exposed to sublethal doses of herbicide flower later in the season, and they flower less. Okay, and so this is what you can imagine just broadly could happen to our weed population resources for pollinators. And so I started this work with Dr. Thielen Ashman and Dr. Vera Iriart. At the time, they're both at University of Pittsburgh. Vera has now started a postdoc in my lab. And one of the projects that Vera did was we collected a bunch of seeds from different weed species from Kentucky and Tennessee, put them in the greenhouse and exposed them to dicamba at a drift level, at a low dose. And we wanted to assess, is there any change in flowering time as well as flowering duration? And so on the y-axis here, I'm showing you each different weedy plant species we were working with. And on the x-axis of the first plot, I'm showing you the difference in the day of first flower. So these are contrast statements. And effectively, the way you read it is, if for day of first flower, if we're on this side above the 0 and we're red, that means that that species flowered later when it was exposed to dicamba drift. Okay, and so what we found is that species responded differently to the dicamba drift in terms of day of flowering. Some responded later, one species responded earlier, and some species weren't affected at all. We did have a pretty significant effect in terms of the difference in flowering duration. So across many of these species, we had a reduction in the time of flower duration. Okay. All right, so this work we did in the greenhouse, which is great, but it's in the greenhouse where you can't really map on how pollinators are going to interact with the plants, right? So today I'm going to tell you about a study that we did out in the field looking for the impact, or looking to see if dicamba drift impacted plant pollinator interactions. That's the first thing that I'm going to talk about. And then I'm going to segue into talking about something surprising that we found from one of our species, where it looks like the species was overproducing in terms of growth and seed number when it got exposed to dicamba. Okay. So the first question, does exposure to dicamba drift catalyze novel plant pollinator dynamics? And so our study design, effectively, we took all of these species as seed from agricultural fields. I'm not going to read the species off, but you can see that we have a couple of morning glories, oxalis stricta, dandelion, Queen Anne's lace, plantago, just really common weeds of the southeastern US. I put together this talk and I realized like, oh, we're probably not going to have the same weeds necessarily. Okay, and so we took individuals, we planted them into field plots at the Mathai Botanical Gardens in Michigan, and we had 4 field plots that served as controls and didn't receive anything. And then we had 4 field plots that were exposed to 1% dicamba. So we planted the plants, we let them grow for a few weeks, and then we sprayed them with dicamba, and then came back and took some data. The data that we focused on was first the abundance of insect visitors just in each plot. So we did 12 rounds of 20-minute surveys at each plot, where we would say like, okay, is there a pollinator here on a plant, flying by, whatever. We also did a typical plant pollinator visitation data, where we looked for both, is an insect approaching a flower on a plant? And that's just like the insect is interested in the flower but doesn't land or forage. And then we also wanted to know, how often are they visiting? And visits where the pollinator went up to the flower, foraged around, and definitely got some nectar or pollen. Okay. And so we again did like 12 rounds of 20 minutes at each of our plots. And that's kind of typical for pollinator observations. And then we also wanted to understand what was happening on the plant side. So we took estimates of size of each of these plants 3 weeks after they were exposed to the dicamba drift. And then we also took estimates of the proportion of leaves that were damaged. We measured or recorded flowering day and then flower number over the course of the experiment. Okay, so plant data as well as plant pollinator interaction data. And some results. So in terms of insect abundance, we found that it declined significantly in the plots that were exposed to dicamba drift. Okay, and this change you could see, so I'm not an entomologist. I know many of you in here are, and that's wonderful, but I appreciate insects. I don't really know what the species are. Like I know ants, I know bumblebees, I know honeybees, right? But that's okay. We're working with a plant pollinator person on this work. So that's a way for me to explain that we put them into functional groups. Okay, so if we saw an ant, we would mark ant, or fly, or bee, true bug, wasp, bumblebee, honeybee, beetle, or butterfly. And so we could see across most of these functional groups of insects, there was a decline in their abundance. Okay, and that's just like who was in the plot. In terms of plant traits, we found that plant traits were altered by dicamba drift. Again, I'm showing you the same kind of contrast plot where I've got species on the y-axis, and here I'm showing you difference in 3-week plant size. Anything in red, any species in red, basically those plants were smaller if they were exposed to dicamba drift. Most of the species showed damage in the presence of dicamba drift. I mean, all the species did. In terms of day of first flower, the majority of our species flowered later if they were exposed to dicamba drift. And in terms of number of flowers, we had some species that produced fewer flowers in the presence of dicamba drift compared to others. For each of these plant traits, there is a significant treatment by species interaction, which basically means what you see here with your eyes, that the species were doing different things from one another when exposed to dicamba. So not all species are going to respond in the negative, right? Okay, in terms of visitation, I'm showing you here the approach rate, the rate of approaches where a pollinator would just come up to the flower and fly away. We didn't see any significant differences in approach rate. We did find for 2 species that there was a lower visitation rate. This is cardiospermum. This is Queen Anne's lace. And so effectively, again, a treatment by species interaction for pollinator visitation, driven mostly by 2 species. Okay, so we collected this data on plant traits and on plant pollinator interactions. I wanted to understand how dicamba, remember we sprayed the plants when they were like 3 weeks growth, just once, a low dose, like 1% dicamba drift. So how is it that these effects are carrying on, at least for some species, all the way to the plant pollinator interaction stage? So to understand this in I don't know, a way that made sense, I developed a structural equation model. So this is where you develop a hypothesized model for the way that you think your traits are interrelated with one another. And so you kind of, if you're not, if you haven't done structural equation modeling before, it's basically you take your biological knowledge of how things interact with one another, and you also hypothesize, okay, I think number of visits is going to be driven by the plant size, the number of flowers a plant is producing, the number of approaches that it gets from a visitor, the amount of leaf damage it shows, and the day of first flowering, right? So you develop this hypothesized model for the interrelationships between all of your variables, and then basically do a series of linear regressions to assess the strength of whether the relationships are there or not, and the strength of those relationships, okay? And so this was our attempt to holistically understand how dicamba drift early on in the season could then affect something indirectly later on in the season. And so I'm just going to give you kind of an overview of what we found instead of throwing a bunch of regression coefficients at you. Anything here, so this is a summary of us making a structural equation model from the plants in the dicamba environment or the control environment. Anything that's a line that's present in black means there was a relationship in both environments. Okay, and so like this relationship here, what this means is that plants that were larger had a larger as a positive relationship, although I'm not showing you that. Plants that were larger had more approaches by pollinators in both environments, whether it was the control or the presence of dicamba. Okay, and so anything in black, the relationships were the same between the different types of data we were collecting. Anything that's in blue is just the relationships were present only in the control environments. And so we can see in the control, the number of flowers a plant produces or the number of visits a plant gets is influenced by the number of flowers it produces. And this was a positive relationship. So if a plant produces more flowers, it's going to have more visits. Okay, and that's kind of a relationship you would expect, right? It was present, however, in the control only and not in the dicamba present environment, which is indicated here by the yellow line. Okay. The other relationship that I think is important that was disrupted by the presence of dicamba was the size at 3 weeks and the number of visits. So in the control environment, the larger you are, the more visits you get from a pollinator. Okay, this relationship was disrupted in the presence of dicamba. Okay. Okay, I wanted to zoom into one species. So this is Cardiospermum halicacabum. It's not a common weed, but it's definitely across some fields in Tennessee. It's a weedy excuse me, it's a viney weed. Definitely looked to me like a horticultural escape. But here I'm showing you a relationship between number of flowers an individual would produce and the number of insect visits. And what you can see is your typical expectation for the way plants and pollinators work, which is the more flowers a plant produces, the more visits it will get, right? There's a positive relationship there. In the presence of dicamba, that relationship was disrupted. It's no longer a positive relationship. And it's even kind of surprising that plants that were producing a lot of flowers weren't necessarily getting a lot of visits. And to me, that suggests that something else could be going on, like maybe the dicamba is changing the volatile organic carbons or changing the scent profile of the plants. It could also be that the plants that were in the dicamba drift plots are just getting lower visits because there's fewer insects there, right? If you're interested in the change in visits in the controls versus the dicamba plots, I just wanted to plot that out. So we can see these are visits over time. The top is the control, and you can see the number of insect visits is much higher in the control compared to the dicamba-only environment. It drops off precipitously. And we can also see, what I wanted to point out is that bumblebees are present in the control environment, but they really are not present in the dicamba plots. And we're finding this across a bunch of other studies too, that bumblebees just really do not like plants that are exposed to dicamba, even at drift levels. Okay, and so the next steps for this project, we're now working on this thing called the FACTS project, which stands for Pollinator Health, Assess, Construct, Test, and Survey. I'm working on this with Jen Blesh, David Sherman, Liz Tibbetts, Noah Webster, and Louis Simon. The general idea is to do kind of what we have already done, but this time focus on prairie plants, or plants that would be in a prairie situation, and assess if there are particular prairie species that might be more tolerant to dicamba than others, and might be producing good quality nectar and pollen even in the presence of dicamba. We want to take a whole bunch of data, use machine learning to construct plant communities that can be optimized for pollinator health, and then take those and translate them, hopefully, into prairie strips. Prairie strips are being used in the Midwest as a way to increase pollinator habitat around agriculture. And so the idea is that we could, design prairie strips that might be tolerant of dicamba drift and deal with some of the negative impacts of the use of this technology. We're also working with Noah Webster, who's a social scientist, and he and Jen Blesh are going to be surveying farmers across Michigan to understand if they're interested, if they would use prairie strips, and to understand their patterns of a use of dicamba and another synthetic auxin herbicide 2,4-D. Some of our initial preliminary results of this work, this is Luc Collier, who is a PhD student in David Sherman's lab, so they're analytical chemists. And Luc has developed a way to detect dicamba in both nectar and pollen by LCMS and or mass spectrometry. And what I'm showing you here are different species. This is some preliminary work that we just got in the summer of 2023. So I'm showing you milkweed, common morning glory, oxalis, and Citus spinosa. Those blue dots are the controls. We don't have any dicamba in the control plant's nectar. But in the nectar of the plants of those species that are exposed to drift levels, again, we're doing 1% of the field dose, we've detected dicamba. Okay, and so the next step is to understand if when pollinators are fed dicamba at these concentrations, does it impact their survival or their behavior? And that's something that Liz Tibbetts, who is on this grant, is doing. Okay, good. All right, so with that, I want to transition to this question: how do sublethal herbicide exposure shape the expression, cost, and evolutionary potential of overcompensation in weedy plants? Okay, and so this really comes out of an unexpected result of that previous study that I just showed you. When we were doing some preliminary work for that previous study, we were looking at day of first flower, total number of flowers, average number of flowers per day, peak flower number. And in one of our iterations of the experiment, we did this multiple times, we found that Oxalis stricta, so this species here, outperformed the controls when they were exposed to low doses of dicamba. And I kept kind of seeing this on and off, and I was like, this is weird. How is it that you expose a plant to something that should hurt it, and it actually outperforms just a normal plant? Well, this is a thing. So this is a thing that's been in the weed science literature for a bit now, and it's called hormesis. And it's just in ecotoxicology and toxicology in general. Hormesis is the stimulatory effect of a subtoxic concentration or dose of a chemical compound that is toxic at higher levels. And so I'm showing you here glyphosate hormesis in the weed Sonchus that's been exposed to different levels of Roundup. So here's the control growing in a pot. It's much smaller than these potted plants that have been exposed to low doses of glyphosate. And then here, of course, you have the negative effect at the dose that's close to the field dose. So I thought this was interesting. And it reminded me a lot of this story from evolutionary ecology, which comes from Page and Whittam back in the 80s. And so effectively, they were studying overcompensation, which is when they found that when mammalian herbivores removed 95% or more of the above ground biomass of Ipomopsis aggregata, relative fitness in terms of seed production and subsequent seedling survival averaged 2.4 times that of uneaten controls. So this was really interesting. When they first published this paper, it was a little controversial because people didn't believe it. They tried to replicate it, it didn't replicate. Now there's been many years of people studying this dynamic. Here's a meta-analysis from Garcia and Eubanks showing that across a bunch of different types of species, whether you're biennial, annual, monocot, dicot, woody, herbaceous, there is an overall effect of overcompensation when plants get a little bit damaged. Okay, so this was a lot of damage, like 95% of the plant being chopped off is huge, but we see overcompensation even at lower doses or lower levels of damage. Okay, so what overcompensation, the study of overcompensation in the evolutionary ecology literature has also showed us or developed is effectively a way to think about the evolutionary dynamics of overcompensation as a trait. And it's really associated with what's called tolerance. Okay, so I'm going to explain what I mean by overcompensation, undercompensation, and the concept of tolerance really quickly. All right, so I'm showing you here a G by E plot where I've got multiple maternal lines. So these are just separate genetic lines. And I'm showing you fitness on the y-axis. We've got control plants and herbivory plants, plants that if you're in a situation where you are undercompensating, that means that you are doing worse in the face of damage than in the control. And so here I'm showing you that there's undercompensation, but there's no G by E. Those maternal lines that you see are doing the same thing. So there's no genetic variation for this trait. In this example, I'm showing you all the maternal lines are doing exactly the same thing. And they're also showing effectively no damage. So if you have the same level of fitness in the damaged environment compared to the control environment, that means you're completely tolerant to whatever it is that's damaging you. Okay, so that's the definition of tolerance. You're able to maintain your fitness regardless of being damaged. Overcompensation, so back to the concept of overcompensation, is where if you were to get herbivorized, you would overproduce compared to the control. So in this scenario, we have overcompensation, but those maternal lines are all doing the same thing. So there's no genetic variation for this overcompensation trait, okay? Here I'm showing you a mixed response where we have some lines that are overcompensating, some that are totally tolerant, and some that are undercompensating. And so what this means is that, well, we have a mixed response, but we have a G by E. So there's genetic variation. The maternal lines vary in how they're going to respond to this damaging agent. Some are going to be completely tolerant, some are going to be undertolerant, and some are going to overcompensate. Okay, so this basically I just summarized for you about 20 years of the evolutionary ecology literature on tolerance. But I felt like we could take this concept and apply it to the study of hormesis, right? So in the weed science literature, people are interested in hormesis and are wondering if it's something that could actually contribute to the quicker evolution of herbicide resistance. Well, for that to be the case, there has to be genetic variation underlying this ability to overcompensate. Okay. So enter my student, Anna Sobel. She was a master's student. She was working on oxalis to look at this potential overcompensation. And so we wanted to ask, is there overcompensation in terms of growth or reproduction in the field? We wanted to redo the studies and replicate. We wanted to ask, is there genetic variation among maternal lines for this hormetic response? And is there an ecological or physiological cost associated with hormesis? Okay, so that's Anna in the field collecting her data. And so this is just a close-up of the species. And this is completely gratuitous, but this species is interesting because it has exploding capsules. See how it like throws its seeds out when you touch it? This is really cool. It's not cool if you're trying to collect seeds or fruits, unless you wear glasses like Anna did, so it was okay. There were seeds and eyeballs though. Okay, so quickly for our study design, we did 2 different field experiments. The first one, we took 31 maternal lines, planted them within 2 treatments and 2 blocks. We had 4 replicates per maternal line for about 500 individuals. For the second, we wanted to repeat it over time and see if we got the same responses. We had in the second field experiment fewer maternal lines, but it's the same basic design. And so here's just an example. We germinated things in the greenhouse, planted them out into the field, and the data that we collected were flowering day. Okay, so same thing. We planted them into our treatments. We let them grow for about 3 weeks, and then we sprayed the dicamba exposed plots with the 1% of the field dose, and then came back 3 weeks later to look at growth and size. We also measured flowering day, the number of leaves the plant produced, the number of flowers, and the number of seeds in the first experiment. And we learned, like every time you do an experiment, you learn things. We learned that one plant produces millions of seeds. So in the second experiment, we counted number of fruits because counting millions of seeds was a bit much. Okay, and so here's our data. Effectively, we're asking, is there overcompensatory growth or reproduction in the field? So for field experiment 1, we found that growth in terms of number of leaves produced was significantly greater in the drift environment compared to the controls. We didn't find any difference in the day of first flower. We did find a very significant increase in the number of flowers that the plants produced when they were exposed to this low dose of dicamba, but we didn't find any difference in terms of seed number. In the second experiment, we didn't find any difference in growth. What we did find is an increase in the number of flowers, but only in the first month after exposure. Over the course of our field study, we didn't see an increase overall in the number of flowers. However, we did see that was enough in the first month to kick up and have a higher number of fruits in the presence of dicamba compared to the controls. And so basically what we're seeing is that, yes, we have evidence for overcompensation in this species in terms of growth, but it's going to change across years, which is probably due to changing environments, right? So it kind of blips on and off. Our second question was, we wanted to know, is there genetic variation for this response? Because if there is, we could potentially have a situation where hormesis or overcompensation is selected for by dicamba drift, meaning that the species could evolve higher levels of resistance probably more quickly. And so we asked, is there genetic variation in any of these traits that we measured? And across all traits, no. We found no evidence of genetic variation for any of our traits, right? And so that effectively means that what we're looking at is just a plastic response, and one that we wouldn't expect would respond to selection or evolve. Okay, and so we also wanted to assess, is there any potential ecological cost associated with overcompensation? Well, if overcompensation, if there's no maternal line genetic variation, there's not really that much reason to look for a cost, right? But what we did notice is that there were changes just given the exposure of dicamba to the width of the flowers. So we measured lots of flowers and found that in the presence of drift, plants produced smaller flowers. And this could be an ecological cost if it translates to less interest from pollinating insects, right? We wanted to see if there was a changed relationship in terms of the allocation a plant was making because of dicamba to flower size versus flower number. And what I'm showing you up top with the blue line is just that there's a positive relationship in the control plants. Bigger plants that produce more flowers are going to produce bigger flowers. They're probably just bigger plants, right? This relationship was the same in the presence of dicamba. So dicamba did not change the per flower allocation of carbon, right? This is just to show you that the smaller flowers were present across the month in which we measured it. So it was kind of a stable change over at least that 1 month. And we found genetic variation in this trait, interestingly enough. Okay, so there is a significant G by E. The maternal lines did different things in terms of their allocation to flower size in the presence of dicamba compared to the controls. We don't know what this means yet or if it means anything. Okay. Okay. So to quickly summarize these two studies, first, we were asking, does exposure to dicamba drift catalyze novel plant pollinator dynamics? We found yes, especially for a couple of species. We found that the presence of dicamba reduces the abundance of pollinating insects. And then for our second little study here, asking, do sublethal herbicide exposures shape the expression, cost, and evolutionary potential of overcompensation in weedy plants? We did find evidence for overcompensation in this species, but it was a very plastic trait, probably responding to different environments between years. There's no genetic variation or G by E for overcompensation in this species, which is, kind of a relief, right? We don't know if that's going to be the answer for all weedy species that are getting exposed to sublethal dicamba drift that overcompensate as a result, but that's what we found for oxalis. And then we also found this interesting effect where the presence of dicamba makes the flowers smaller. They produce more flowers when they're exposed to dicamba, but they're smaller flowers. Okay. So that's all that I have to present today. So I just want to thank the people that I've been collaborating with and working on this with, people from my lab as well as funding sources, and the people that I have outlined in blue. This is the people whose work I'm going to be discussing today and tomorrow. So with that, I'll be happy to take any questions. Yeah.
So awesome talk.
Thank you.
In terms of the first part of the talk, we talked about these more sublethal doses, noticing a lot of kind of population level variation.
Mm-hmm.
And so I'm wondering, in terms of the mechanisms of evolution of resistance to a lot of these pesticides, do you think that there might be kind of these soft selective swings going on?
Yeah.
Like not necessarily a reduction in census population size, but there are individuals that are doing better. Maybe like drift and these sublethal doses are kind of a mechanism for resistance.
Yeah, no, that's a great question. Yes, and definitely low dose exposure to herbicides can lead to the evolution of resistance, but typically using different genetic mechanisms like detoxification, polygenic responses, whereas the field doses typically will highlight resistance from single genes or particular alleles. But definitely some of the work that Paul Neve has done has shown that low dose selection can rapidly increase resistance. And I think he did that in Lolium rigidium when he was in Australia, but then also he was working with an algae and did it in the lab, which was pretty cool work. Yeah. Yeah.
Thank
you.
Many of those plants, they are weeds, right?
Huh.
They are weeds.
Yeah.
And they are naturally assumed to be very resistant to stress. But we all know that resistant intolerance, when we're talking about the trade-off, they can be affected. They will be affected by, let's say, nutrients, drought, and other environment factors. I'm just curious how these other environment factors will affect that G by E tolerance or resistance of those weeds when play together with
the overcompensation.
What's the name of the insecticide? I forgot.
Dicamba?
Dicamba, yeah.
Yeah, no, that's a great question. And that's something that we should probably look at is, how tolerant a species really is when it's exposed to multiplicative types of stress. I have a student who's working on interactive effects of herbivory and herbicide, but in other types of stressors like heat or lack of water, we haven't looked at quite yet. Although we are doing, it's a great question. I don't know is the answer, but we do have one experiment going on right now where we're working with mutant lines of morning glory, and we've put them out there in the field, and we're exposing them to herbicide and also overwatering, and trying to see if there's any kind of change, according to whether they get supplemental watering or not. But yeah, no, that's a great question, and it's not one that really has been answered. People are talking about it, but yeah. Any other questions? Yes, go for it.
I'm fine, the bean. Okay. Yeah, just the cook since how many days? I know cook, I need transfer my food now. I like eat my food, put my food on my freezer. And then that freezer, your food will just spoil if you put food inside. Another food in our food, they always make mistakes with that. Now they put hot food inside food.
Yeah, so that's a great question too. So we have had a small sort of side project where we had an intern one summer who was interested in figuring out what species of insect they were looking at, and found they were looking at like beetles and ground dwellers and doing, catchments or whatever, and trying to figure out what species were there. But they found a reduction in overall presence of insects just in general, not necessarily just pollinators. But we haven't, it's the kind of thing where we looked at it preliminarily, but haven't gotten to it in depth yet. Yeah.
At home, I beg, send me, I know people say you don't do this for it, but send me templates for weakness.
No, we haven't. That's a great question. Yeah, we're doing some microbial. So Vero, who's now working in the lab, is doing some 16S microbial sequencing, as well as some fungal sequencing to see how dicamba disrupts that. And she was thinking like, well, maybe I should look at patterns of herbivory on the roots as well. But that's as close as we've gotten so far. Yeah.
Just send me the pure.
Other questions?
Well, I mean, make sure you need
responses, negative, positive, the gamma.
Like name like thank you the PDF.
Composition, or since some you expect these plants that have a positive response to become more abundant over time or in places. Do you have any longitudinal or
I wish. Yeah.
Sites have that happens. You see you shift it.
That's a great question. Yeah. So one of the things that why we're interested in overcompensation or hormesis is because maybe a hormetic response doesn't evolve, but maybe it underlies or somewhat underlies weed shifts. And weed shifts are this phenomenon where you have weeds that can be, just sort of naturally tolerant to different herbicides. And so if you come in and use a new herbicide, you would expect those weed species that just kind of can naturally deal, they're not evolving higher levels of resistance or tolerance. You would expect that community would become predominated by that particular weed species over time, just through ecological filtering, not necessarily any type of evolution. But yes, I'm interested in the idea that hormesis is contributing to weed shifts. And we started our work back with TLN in 2018 and have estimates of like what species are in particular fields, with the idea that we would get funded again and go back and see if there has been any sort of weed shift. But yeah, that's kind of in the works, but we haven't done it yet. Yeah. Good question. Okay, if there are no other questions, Thanks for being here. Oh, is there a question? Someone say something? No? Cool. Okay. Thank you for listening to my talk, and I'll be giving another talk tomorrow.